yes, I have plenty of time to see. Oh. All right.
You went over.
Oh. Oh, okay. Yeah. Oh.
Passing out individually?
What's that? Oh, I've been meaning to ask about the fish. Oh, okay. No. Right. What's that? Okay. The fish. Somebody said they were, there are 6 fish.
Who's that other Rochelle? She was going to want a goldfish?
Yeah.
She wanted a couple that she's not here today.
Oh, okay.
Take them home? I
Um,
don't know her name.
tomorrow. If you want, fish. Yeah. Goldfish. Alright, what's that?
Eat the last beta fish that we got. Well, didn't really eat it, just kind of played with it a bit, just ruined it. My son took my last beta fish and shoved it in his pocket. Tried to run off. I was like, oh my God.
What are you doing?
Once.
She was had her hand in the bowl was petting it. I'm like, please don't pet the fish.
Okay.
Come to you.
Maybe we'll do a carrier first. Later on, cut out chromosomes and see any disorder. Trisomy 21. When there's 3 chromosomes on the 21st pair, where there should be only 2 chromosomes. If you see 3 chromosomes on the 21st pair, what's that? Down syndrome. Certain, if it happens, multiple chromosomes on a specific pair, these larger chromosomes, most likely that would result in a miscarried offspring. But these smaller chromosomes, it's sometimes passing through the tongue. And again, Down syndrome, either trisomy, it's known as, 3 chromosomes on the 21st pair, instead of a homologous pair. Usually that does not result in a miscarriage. But anyways, somatic cell history. Every single cell in our body, when we get cut, or repair, or for growth, right? Except for a few, right? Or known as somatic cells. Our sex cells will not undergo mitosis. but every single somatic cell in your body will go through these two main phases, interphase and mitosis. Again, only sex cells will enter the meiosis, and that will deal with meiosis next week after the exam. The first main thing of a somatic cell history. Actually, this would be a good essay question, short answer question. Explain the cell cycle of a somatic cell. And you would need interphase. Right? 90% of every single somatic cell in your body is in interphase. It's only when the ANCA gene is activated will it turn into from interphase to mitotic division, mitosis. Perhaps you're familiar with these stages of mitosis, prophase. Some people, and it depends on who you talk to and what textbook you read, throw in between prophase and metaphase, pro-metaphase. I started using pro-metaphase too.
In addition to both prophase and metaphase, in between the two, or are you just combining them?
Yeah. Some people, textbooks and authors use prophase, metaphase, anaphase, and telophase. Other authors or textbook throw in pro-metaphase. That's in between metaphase, meta. What is meta refer to? Middle. Meta, in the middle. Okay? So that's where all the chromosomes in the duplicated form, right? In the duplicated form, or in the middle of the cell. It's amazing that how many students get this wrong on a lab practical and or on an exam. Right? When I ask what phase is all the alignment of the chromosomes in the middle of the cell, and you would say metaphase, middle, meta. These are all the chromosomes. In a whitefish blastula, we're going to look at eggs of a blastula, a fish, for the animal cell. And for the plant cell, we're going to look at allium, or the onion tip, the tip of the onion root. The onion, these are plant cells, right? Very, very rigid cell wall. Again, these are animal cells, primarily the whitefish blastoid. My main point here is that only sex cells will undergo meiosis. Every single cell in your body that's not a sex cell is undergoing mitosis. Mitosis is just a carbon copy or a clone of the parental cell. Yes? Jen?
You said, so anything non-sexual is somatic. Is that for all animals and plant cells?
Yes.
Okay,
Right,
but
yeah.
some plant cells have,
The gametes,
Yeah.
right.
Right.
Particularly, we're gonna deal with angiosperms, flowering plants. So the flower, most flowers are bisexual, most ornate flowers. When I have two sisters. When a guy that I did not like gave one of my sister's flowers, I would pollinate all of her flowers. And by pollinating the flowers, they'll immediately, within a day or two, senesce and die.
even if you water them?
Oh, right, yeah, because once a flower is pollinated, it's done
It
its
doesn't have
job.
any more purpose,
It's
yeah.
right, so by pollinating flowers, and devoting all of that energy into the developing embryo, right, or the developing seed. But if I liked the guy, I would castrate those flowers. By castrating the flowers, I would prevent, and florists do this a lot, they will prevent pollination to occur. So those flowers will stick around for a week or two.
How do you castrate a flower?
Right.
You know how to pollinate, but.
Right. Now that, okay. Most flowers, the female portion, and the male portion. The anthers, and the pistil. The pistil is the female portion, and the male portion is the anther. These fuzzy. The anther is supported by the filament. Together they're known as stamens. That would be the male portion. But the pollen will be produced by the anther. Okay? The anther or the pollen is the male gamete. Right? So by snipping off the anthers, you're preventing the pollen from hitting the style. These are part of the pistil. It's also known as the carpel. But we'll talk more about that. But that's how you castrate a flower. most flowers, ornate flowers, or nice looking flowers, are bisexual. But anyways, sex cells, the pollen will be produced by the anther, and those would be a removal of half of the homologs. And again, those would be done, that would be done by meiosis. And those would be sex cells in a plant. The animal sex cells produce the egg and the sperm. The female, the egg, and the male, the sperm. The animal's somatic cells will undergo, you don't have to worry about half the number of chromosomes being put in your gamete. So in essence, mitosis is, barring any mutation, a carbon copy of the parental cell. There's another term for
You think the gamete is the carbon copy?
no. Gamete is produced by, all gametes are produced by meiosis.
So the meiosis is
Half,
basically making the carbon
right,
copy.
half
Okay.
of the homologs. Mitosis is a carbon copy of. They're known as diploid cells. A diploid cell is this right here. Everything, diploid, di. Ploid refers to, ploidy refers to the chromosomes. Okay, so ploid would refer to the chromosome. Diploid cells, or diploid cells, have homologous chromosomes. One we received from our mother, one, the other member, we received from our father. Again, they are in any diploid cell. The second pair of homologs, or in a diploid cell, is again, one we received from our mother, the other one we received from our father. Diploid cells, 2N. 2N. N would be the chromosomal number of that species. In our species, our N number is 23. So our N in humans equals 23. So 2N would be a diploid cell. And every single diploid cell will undergo mitosis. Mitosis uh, 2N. So again, where did this other cell come from? It just magically appeared from the, this is a diploid cell. So think of this as in here. So I could see right in here. So where did this other cell come from?
Because the pair split.
Ooh, on the contract.
Duplicate.
Before, right, yeah. Before they split, any split, what has to happen to the chromosomes? They have to duplicate. So therefore, this is a duplication. So there's during interphase, before any mitosis, primarily there's three parts, main parts of interphase. The second part of interphase, known as the S phase, this is where the DNA will replicate and produce duplicated chromosomes. So again, you need, before mitosis even can be thought about, right, you need duplication of chromosomes to produce half, right, well, a carbon copy of each other. Right? Again, these are known as sister chromatids. Together, these four sister chromatids make up what is known as the homologous pair from the unduplicated chromosome. So the S phase, the S phase of my interphase, the second part of interphase, we'll see the duplication of chromosomes. Yes. Oh. Interphase, there's three main parts, okay, or subparts. GAP1, G1, S phase, replication of DNA. So the DNA will replicate, and again, producing a centromere with two sister chromatids. And again, barring any mutation, those sister chromatids are exactly alike. And then followed by GAP2. This tubulin, what macromolecule would tubulin be? Right, a protein. Whenever you see IN at the end of a word, in biology, it's a protein. Tubulin, very, very important in cellular reproduction. I'll go over that. Interphase. These GAP1 and GAP2, before the electron microscope, biologists knew that there was something going on, but they couldn't see it. Okay? So they called it GAP. Okay? And then they saw under a compound microscope, chromosomes appearing. Okay? And they knew they could see that. Okay? So they named that the S synthesis phase, the synthesis phase, or the S phase. You can see the chromosomes. They didn't know that there was DNA in there. There were factors, early biologists. But anyways, and then they knew that there was something else going on. But again, after this synthesis, where the chromosomes were being formed. And then they couldn't see that, so it was known as a second gap in this interphase. So that's where you get these G1 and G2, or gap 1 and gap 2. But now, with the electron microscope, you know exactly what. Okay, mitosis, 4 or 5 phases. 4 or 5. Some biologists have a 6-phase cytokinesis. You could add that.
It's on the study guide you put,
What?
the little study guide that you gave us, that was great, that one has the cytokines.
Yeah, kinesis is to move. Kinesis is to move. Kinesis tape. Cyto is again the cell. The actual splitting of the daughter cells. That's where. Okay. Diploid cell. Meiosis. Meiosis is you are starting with a diploid cell. And you're producing 4 haploid cells. Haploid, half the number of chromosomes. Again, not a random halving of the number of chromosomes, but the removal of one member of each homolog. But again, we'll deal with meiosis after the next week. But I'm all set with this. Diploid and haploid cells. Haploid is half the number. Ploid is again the chromosomal number of half of the. But mitosis. Mitosis, again, every single mitotic division begins with a diploid cell and ends up with 2 daughter diploid cells. Again, barring any mutations, both of those daughter cells are clone copies of the parental cell. Mitosis, multicellular organisms. Again, somatic cells. Right? Mitosis is for somatic cells. Only when you're producing gametes would you, the sex cells will produce gametes, and those gametes are produced by meiosis. Right? But every single somatic cell will undergo mitosis. The chromosome structure, again, the DNA is wrapped around specialized proteins. The centromere joins the sister chromatids. Again, those are exact copies of the DNA would be. The microtubules will attach to the centromere during metaphase. Chromosomal number. Amoebas, dogs have 78 chromosomes. Humans have 46 chromosomes, or 23 pairs. Amoebas have 50 chromosomes, or 25. The intelligence of any species does not give you the chromosomal number. Right? When I had my dog, he was smart, amoebas have, the chromosomal number will give you the adaptability, right? How that organism is able to adapt to a varied environment. Right? The more chromosomes any species has, it refers to the longevity that that species. We are such a young species, as opposed to all these other species around. And that's one of the main reasons why we have so few genes or so few chromosomes. The adder ferns